So, hello and namaste to all of you. I'm Chalu Singh from Department of Physiotherapy and Diagnostics from Jyoti Vidyapeeth Women's University. For today's lecture, I'll be talking about osmotic solutions or tonicity of solutions. So, in last lectures, I've told you about osmosis, its various mechanisms that are acting on osmosis, its principle, the factors affecting the mechanism, and various examples related to it. And also, I've also discussed in last lecture about osmotic pressure. Before going through the tonicity of solution, let's see what is basically osmotic pressure. Osmosis basically, first of all. So, osmosis, it is a biophysical process occurring commonly in your biological systems where solvent molecules move across a semi-permeable membrane towards a region of high solute concentration, and it is a type of passive transport and is directed towards the direction that tends to equalize the solute concentration across a semi-permeable membrane. And it is a common process, so taking place in most of the biological membranes in the organisms. So it in biological system, the solvent mostly is water, and however, osmosis can take place in other liquids and even in gases. So the principle behind osmosis lies that the two vessels, they are separated by semi-permeable membrane, and the solutions are added to both solvent or solutions that are added to both the sides of the towards the both sides of the membrane. So the and when solute is added, so the movement takes place from higher concentration side to the lower concentration side. So, and if external pressure is exerted, this transfer process, it can be stopped or it can be reversed. That is your reverse osmosis. And the pressure that is applied, or the pressure that is minimum pressure that is necessary to stop the solvent transfer, is your osmotic pressure. And uh, further, the various mechanisms which I've told you, that the mechanism that is acting upon in order to osmosis to take place is your driving force that is allowing the movement of your solute particles from a higher concentration to a lower concentration gradient. So that is basically your concentration gradient that is providing the movement from one side of your membrane to other side of the membrane. And nextly, I told you about osmotic pressure, and about I told you that the formula of osmotic pressure is pi is equal to mRT, or pi is equal to ICRT. Or nextly, for today's section, let's start about tonicity of solutions, or various osmotic solutions I'll be talking about. So, what are the various what is basically the term tonicity mean? So, tonicity is the ability of extracellular solutions to induce the movement of water in and out of a cell by the process of osmosis. So, the tonicity of a solution is determined by the concentration of solute and solvent molecules in the solution. So, on the basis of tonicity of solutions, they are termed as hypotonic, hypotonic, and isotonic solutions. Now, first of all, let's discuss what is hypotonic solution. So, if an extracellular solution has less concentration of solute, then that inside the cell, the solution is termed as hypotonic solution. Or when a cell is placed in hypotonic solution, the movement of water, it occurs into cell and resulting in endosmosis. So endosmosis, that is movement, will take place from outside to within the cell. So it will cause swelling of the cell. So the cell in such a condition will swell up, and if continuous swelling will take place, it might burst. Now, uh, let so in hypotonic solution, uh, it has a lower concentration of solutes than other solution. So in biology, a solution outside the cell is called as hypotonic if it has a lower concentration of solute relative to your cytosol. So due to osmotic pressure, water diffuses into the cell, and the cell often becomes turgid and or bloated. So for cells without a cell wall, such as in animal cells, if the gradient is large enough, the uptake of excess water can produce enough pressure to induce cytolysis or rupturing of the cell. And when the plant cells are in hypotonic solution, the central vacuole takes on extra water and pushes the cell membrane against the cell wall. So due to rigidity of the cell wall, it pushes back and it prevents cells from bursting, and it is called as Turgor pressure. Now, the next point that next type of solution that we'll be discussing today is your hypertonic solution. So, what is hypertonic solution now? It is just the opposite of your hypotonic solution. Hypo and hyper. These are just opposite terms. So, if an extracellular solution it has more the concentration of solute than that of the inside cell, the solution is termed as your hypertonic solution. And when a cell is placed in hypertonic solution, the movement across out of the cells it will result in exosmosis. So water is trying to exit your cell. That means from normal size, your cell will shrink. The cell shrinks down and loses the ability to divide and even function. Now, what is your isotonic solution? So, when an extracellular solution has the same concentration of solute as that of inside the cell, it will be termed as isotonic solution. And when a cell is placed in isotonic solution, basically no movement of water takes place across the cell membrane. And in this case, the size of cell is not influenced and no movement of water takes place. So, it, like in case of hypertonic solution, it has greater concentration of solute than other solution. So, in biology, the tonicity of a solution is usually refers to its solute concentration relative to that of another solution on the opposite side of a cell membrane, and a solution outside of a cell is called as hypotonic if it has greater concentration of solutes than your cytosol inside the cell. And when a cell is immersed in a hypertonic solution, osmotic pressure it tends to force water to flow out of the cell in order to balance the concentration of solutes on either side of your cell membrane, and the cytosol is conversely categorized as hypotonic, opposite of your outer solution. And when plant cells are in hypertonic solution, the flexible cell membrane it pulls away from the rigid cell wall but remains joined to the cell wall at points called plasmodesmata, and the cell often takes on appearances of a pincushion, and the plasmodesmata almost ceases to function because they become constricted at a condition known as plasmolysis. In plant cells, the term isotonic, hypotonic, and hypertonic, it cannot strictly be used accurately because the pressure exerted by cell walls significantly affects the osmotic pressure point. And some organisms have evolved intricate methods of circumventing hypertonicity. So, for example, salt water is hypertonic to fish, so that lives in it. And because fish needs a large surface area in their gills in contact with seawater for gas exchange, so they lose water osmotically to the sea from the gill cells, and they respond to loss by drinking large salt water, and actively excreting the excess salt. So this process is called as osmoregulation. Now, what is isotonicity basically? The solution is said to be isotonic when its effective osmolar concentration is the same as that of another solution. And in biology, the solution on either side of the cell membrane are isotonic if concentration of solutes outside the cell is equal to concentration of solutes inside the cell. And in this case, the cells neither swell nor shrink because there is no concentration gradient to induce the diffusion of large amounts of water across the cell membrane. And water molecules freely diffuse through plasma membrane in both the directions, and the rate of water diffusion is same in each direction. So the cell wall neither gain or nor lose water. So an isoosmolar solution, it can be hypotonic if solute is able to penetrate the cell membrane. And for example, an isoosmolar urea solution is hypotonic to your red blood cells and causing their lysis. And this is due to urea entering the cell down its concentration gradient and followed by water. So the osmolarity of normal saline is that is 9 grams NaCl dissolved in water to a total volume of 1 liter is a close approximation to the osmolarity of NaCl in blood, which is about 290 milliosmoles per liter. Thus, the normal saline is almost isotonic to blood plasma, and that's why we give saline solution to the patient. And here we always have to measure the concentration of salt which we give, so that no osmosis, it's basically the isotonic solution. So neither sodium or chloride ions can freely pass through the plasma membrane, unlike urea. So that will be it. So these were the three types of osmotic solutions, or these were basically three types of tonicity, that is your hypertonic, hypotonic, and isotonic. Now, in next class, we'll study further about osmosis, and until then, take care and do well.